Welcome to a quick tour of DEGRONOPEDIA. This video will give you a brief overview of the features and capabilities of our web server. DEGRONOPEDIA is designed to make your research on protein turnover easier by providing a user-friendly platform to search for degron motifs in any protein and deliver their broad context. Whether you are working in the field of protein degradation or simply want to gain insight into the turnover of your protein by the ubiquitin proteasome system, our server offers a wide range of functionalities, including degron visualization, machine learning to predict protein and C-terminal stability, proteolysis simulations, and a wealth of information about degrons, including their post-translational modifications, mutations, and interacting E3 ligases. We'll gladly guide you through the DEGRONOPEDIA interface and show you how to take full advantage of its potential. So, let's get started. DEGRONOPEDIA accepts three types of queries: UniProt protein ID from one of the selected model organisms, protein sequence in FASTA format, or protein structure in PDB format. Let's run DEGRONOPEDIA using the UniProt ID as an example, since this type of query provides the most comprehensive output. After submitting the query, we have to wait a while, as DEGRONOPEDIA screens the protein for the presence of over 400 degron motifs, and performs other calculations that we can customize on the homepage, but more about that a bit later. Once the calculations are complete, we see a summary panel, including the number of degron motifs in our query and the hydrophobicity scores of its C-terminus. Next, we have a visualization of the found degron motifs overlaid with information on intrinsically disordered regions, solvent accessibility, secondary structure, post-translational modifications, and mutations. The definitions of some of these features can be adjusted in the settings panel on the homepage, such as thresholds for recognizing residues as disordered or buried. We then have panels providing information about the stability of the C-terminus of the query based on experimental data from two large-scale studies covering almost the entire human proteome. Since we have marked the option to use our machine learning models on the homepage, we also get such predicted stability values and their visualizations for easy interpretation. Next, we have a table with detailed information about each degron motif, its location in the sequence, type, and relevant information from the literature. In addition to the degron motifs available on the server, user can specify their own using the appropriate panel on the homepage. DEGRONOPEDIA gathers pre-calculated multiple sequence alignments of orthologs at various evolutionary distances. These, along with those provided by the user, are used to calculate and visualize the evolutionary conservation of each degron motif. Structural information, including disorder and solvent accessibility, is provided for each degron motif, as degron location within the disordered region and high solvent accessibility are clues that an identified motif may act as a degron site. Then we have post-translational modifications occurring within each degron motif, as well as in its neighborhood, as such modifications can modulate degron functioning. We also provide information on known missense mutations, reported likewise for each degron motif and its neighborhood. Next, we present the context of each degron motif according to the proposed tripartite model. In this model, a degradation targeting degron consists of three elements: a primary degron, which is a short linear motif recognized by E3 ubiquitin ligase, a secondary degron, which is a lysine to which ubiquitin can be attached, and finally, a tertiary degron, which is an intrinsically disordered region that initiates proteasome-dependent protein degradation. It has been suggested that secondary and tertiary degrons play auxiliary roles that affect ubiquitin signaling. The absence of one of the elements of the tripartite degron model, such as an intrinsically disordered region near ubiquitinated lysine, may result in a non-proteolytic ubiquitination function. In DEGRONOPEDIA, we provide the full context of the tripartite degron model for each identified motif. Moreover, we consider not only lysine, but also cysteine, threonine, and serine residues as potential secondary degrons, as they can also undergo ubiquitination. Degrons often occur at the amino or carboxyl end of proteins, acting as initiators of NC-degron pathways. Since they can appear after protease cleavage, DEGRONOPEDIA simulates sequence nicking based on user-provided motifs or sites, experimental data, and theoretical predictions and screens for degron motifs in emerging protein termini. Finally, we report on E3 ubiquitin ligases known to interact with the query protein. All the outputs that DEGRONOPEDIA provides are described within question mark icons. We also provide a comprehensive tutorial on the functionalities of our web server. Finally, the list of the available degron motifs that DEGRONOPEDIA uses to screen proteins can be downloaded in the form of an Excel file. In summary, our goal is to provide a tool that will stimulate research on degron signaling by delivering output information in a validation-ready context. We hope that DEGRONOPEDIA will be a valuable resource for your research.